Hello and welcome to the Research in Brief podcast with me, your host, Chris Curwen. Today I'm absolutely honoured to be joined by a good friend to the show and a brilliant scientist, Dr. Milan Mušo. Milan is currently a Postdoctoral Research Associate of Human Metabolic Diseases and Ageing at the Institute of Metabolic Science, University of Cambridge. Milan has recently authored a review of the current literature surrounding the role that RNA plays in the aging process, appropriately titled RNA and Ageing. So welcome, Milan, and thank you so much again for joining me.
Hi everyone, glad to be here.
So I'll dive straight into the questions. For those who are listening who maybe don't have a huge background in biology, can you just explain what RNA is?
Sure. RNA, actually very much similar to DNA, is a nucleic acid. It's a large molecule, and essentially what it is, a messenger. It's a messenger that basically delivers the message from DNA to protein and allows our body to function. But apart just from being a messenger, and that's also what my chapter is about, is it also has many enzymatic functions. It can allow itself, it can have a role. And in fact, there are theories that RNA has been the original storage of information. This is even where life once might have originated.
What is the sort of best current understanding of what causes aging in a more general approach?
So there are obviously multiple theories about this, but I think the best one so far seems to split aging in multiple agings. Essentially, aging is a group of different processes leading to deterioration, dysfunction of our cells, of our organs, and of the whole organism, how everything communicates together. There's been efforts to characterize some of these processes. And for example, the Carlos Lopez paper defined the 9 hallmarks of aging, some of them such as the dysfunction of our mitochondria, the organelles in our cells, the basically loss of integrity in our DNA, epigenetic deregulation, and other processes. So essentially, I think of it as a natural process. Aging is basically exists because we lack proper maintenance mechanisms. So even if we wouldn't necessarily interfere with our organism, we would provide it the best conditions you can, aging would still occur. And we know this, for example, because DNA mutations, such as previously been postulated to be one of the causes of aging, itself aren't the whole story. You can still generate perfectly healthy clones from very old cells and generate a new healthy organism. So we know there must be something more happening. Essentially, to defeat aging, we would have to even upgrade our DNA so it generates better maintenance mechanisms.
So you talked there about genetic and genomic instability being maybe one of the root causes of aging. But where does RNA come into the picture? What role does RNA have in aging?
So RNA, being the key intermediate between the DNA and our proteins, or essentially the function of the cell, is really the point of regulation. So although RNA would directly transcribe the message of our DNA, that's not all of the story. We have multiple genes, 20,000 genes, and each of them in every cell is transcribed or converted to RNA to a different extent. Our RNA, for example, can be present at different levels. By changing these levels, by either driving it up or down, we can affect the biology of organisms. And this is basically what occurs during epigenetic regulation or during some of the other processes we'll talk about. So it occurs on the level of the amount of RNA, but also on the level the RNA is put together and processed. So there is a process called splicing, which you might want to talk about later, basically arranging the different pieces of RNA. So that also can be affected. And we know there are such changes with age, although there are organisms where we don't see this. You might be aware of the bristlecone pine tree, which has been shown to live up to 5,000 years. And there, these processes have been very minimal. But in majority of organisms, these things change quite substantially.
So you've given us a great explanation as to what RNA is and what role it might have to play in aging. But how do we actually observe RNA and gather information on the sequence of our own RNA and the RNA of other organisms?
So RNA basically is quite a large molecule. DNA itself, for example, has 2-strand. RNA has only single strand. And generally in human terms, usually the plan or the plan of a building is usually much smaller than the building itself. In the case of RNA, actually, RNA is often many times larger than the eventual protein is being produced. So we have an advantage. We can separate it by size. And also, as it's in the name, RNA ribonucleic acid is an acid negatively charged, so we can use also chemical methods to separate it. So basically what happens, we can lyse cells, whichever cells we're interested in, or even blood, pull out these species based on the methods I've described. And then multiple methods again. I think the most common one is to use a viral enzyme, a reverse transcriptase that will read RNA back to DNA, and then using standard sequencing approaches or next generation sequencing approaches, we can read the sequence base by base, and getting this information, then we can align these sequences to our genome so we know where it comes from, but importantly also how much of this RNA there is. And then we can monitor changes between different conditions or between different organisms. Of course, there might not be that much known about the organism or the condition of interest. And then there are cases where we would discover new RNA species. So they would align only to some parts of the genome, but not others. And yeah, this is how we would describe so-called transcript isoforms.
So to someone who doesn't know too much about the background of RNA and sequencing, this sounds like quite a complicated process, but is this something that we've been doing in the lab for a while and we're getting much better at?
Yes, I think this is a very old process, and we've been analyzing RNA using traditional methods for probably over 30 years. Basically, we have the ability to look at single RNA species for a long time using, for example, fluorescent methods. So if we know a gene of interest, we can design certain probes that would tell us its levels in a cell. Obviously, what came later was this next generation sequencing, which allowed us to look at almost all the transcript in a tissue or in a sample. We could take a piece of liver from a mouse and sequence that. So those approaches are becoming much cheaper and also more informative and less biased, because we're looking at the whole sample and then only after that looking for the interesting things we find.
So you talk about in several studies that have deployed the use of single cell RNA sequencing in their investigations. And you go on to say in your discussion, and I quote, "RNA sequencing will likely continue to be at the center of future revelations about the aging transcriptome." I've got two questions in this. What are the advantages of being able to sequence single cell RNA rather than whole populations of cells? And what avenues does this single cell sequencing open up to us?
So single cell RNA sequencing is absolutely wonderful technology. In terms of the aging field, for example, we have been noting for a long time that there's a huge variability in the numbers of genes that are changing with age based on the tissue we look at. So people looked at Drosophila, they looked at the whole body and found around 30% of our genes being dysregulated with age. However, then when you look at something like mouse, you would find just a few percent changing. And the same with humans. In human peripheral blood, the percentage is around 12% of gene changing. But when out of that peripheral blood, you only take leukocytes, white blood cells, you get only around 3% of genes changing with age. So already then it was quite clear there's something going on. And perhaps it's not just that the cells are changing their RNA amounts, but actually that there are differences in the cell populations themselves. And this is absolutely wonderful technology. The single cell RNA sequencing allows us to answer these questions. For example, a study of aging pancreas were previously showing that you are seeing increases in some cycling-dependent kinases, basically markers of senescence. But with single-cell RNA sequencing, you basically are able to see that what is actually happening is that there is a rise of these specific senescent cells rather than those genes being affected in that specific population. And this kind of studies have been found in multiple other cells. For example, in other aging tissues, we saw a reduction in cell cycle genes. But actually what we found there was just simply reductions of cells that were dividing. And this was, again, a very specific small population. So where does this lead us? So what single-cell RNA sequencing can do is really distinguish which cell types are aging and perhaps which cell types are more susceptible to aging. And of course, not to say that cell type is a very not particularly well defined statement. There are multiple cell types within cell types, so this boundary isn't that clear. And obviously single cell RNA sequencing will allow us to explore that further. But this is basically leading to studies such as tabula muris, which is a study of aging mice. I think they have around 17 tissues from mice over 6 different time points. This is not a very high cell number, so they haven't looked at that many cell numbers, so the data is a bit variable. But in the future, hopefully there'll be more of such studies and we can pinpoint which tissues are really aging the most. So for example, in this study of the mice, we could see that adipose fat tissue was one of the first tissues to show aging-related changes with increasing inflammation markers showing up. And the other way where this is leading is now most recently spatial transcriptomics, where you can use technologies to basically look at not yet single cell, but groups of cells on a slide exactly at a particular position. And then you can relate RNA levels at a particular location. So that allows you to see, is there some kind of interaction between aging or let's say senescent cells, and are they affecting other cells and other known questions? Yeah, that's going to be very exciting.
Is this technology already affecting your own research? Are you taking on these techniques to establish in your own investigations?
So currently, I'm not working with single-cell RNA-seq data. Unfortunately, I would like to, but those databases are publicly available. Basically, if you have a small question or you want to look at the expression of your own gene of interest or particular project, you can literally go online and look these things up. You can search the cell populations. In my own group, in the IMS, people are very interested in studying the brain and the hypothalamus, which actually regulates energy intake and our sense of hunger and satiety. And definitely there, the interplay of which receptors are in which cells is crucial to understanding human biology. And for example, how we communicate multiple aspects of our biology.
Yeah, no, that's really fascinating. And from my understanding, it really improves the granularity of how we actually look at the cell types in our body. Now, going on to more of a general aspect, what are the key changes that occur in the transcriptome and RNA during aging, and what are the outcomes of those changes?
So there is the general aspect, firstly, which is the change in RNA levels, and this is the messenger RNA, the one that gets converted to protein. Overall, these can be quite different between organisms and tissues, but I think what I describe in the chapter is the genes that are downregulated in aging tissues are generally less similar between tissues. Basically, they're cell type specific often. This would include, for example, I give example of Achilles tendon. There we lose expression of extracellular matrix, the between cells proteins. In neurons or in the brain, often you would lose some of the RNA that's related to expression of genes that guide the axon formation, the growth of the neurons and contact with other neurons. So that tends to be quite specific. However, the genes that are upregulated seem to be more similar, and these are related to inflammation and stress response. But still, looking at overall at all the genes, I think what we find generally, and there are exceptions to this, but generally it seems there is a reduction in, for example, mitochondrial genes, so the energy powerhouses of our cells, the proteins that go towards them from the nucleus, and the RNA that creates those seems to be downregulated. The other one that seems to be going down in many species and tissues is the ribosomal synthesis genes. So basically RNAs that will code for proteins that will then make more proteins. So those seem to also go down. But really about half of my chapter was mainly focused on RNA-based regulation. So what is happening with, apart from just RNA levels, so that there are major changes in RNA splicing, how RNA is arranged actually. And it seems that at least in some tissues, actually RNA splicing can be even a better predictor of age than actually RNA expression. And also what is interesting, that those genes that are affected by RNA levels, so for example, the ones that go down, seem to affect different biological pathways than the ones that are affected by splicing. So potentially these two could be independent mechanisms driving different aspects of aging. There are other things that I describe. For example, generally we see a reduction, that there is an effect on RNA silencing. For example, in senescent cells, also we see effects on the 3' UTRs, the ends of transcripts that can basically cause, generally the longer end of a transcript you have, the faster you get degraded. So we see in senescent cells, those UTRs seem to be generally prolonged. Other things we see, there are these things, circular RNAs. It's not fully known what they are, but they are connected from the start to the end. And those are highly upregulated in the brain. Actually, people think of them as a very great potential biome. In the brain also, we see some changes of nonsense mediated decay. So generally, it seems the RNA quality control seems to go down with age, and especially in neurons. Actually, in worms, there are some studies showing that this decay or nonsense mediated decay might be key to some longevity pathways in worms. All of this I'm describing might be especially important in neurons, which are obviously long-lived and therefore need to have a higher quality control.
So you mentioned RNA silencing there. I was just wondering if you could just tell us a bit more about what that actually is and what role it has to play in aging. And in the sort of processes that you just described, are there any low-hanging fruits when it comes to actually developing any treatments for diseases when we're looking at our RNA?
So basically RNA silencing, and what it is, it's basically when you inhibit the process of converting RNA to protein. So this occurs mainly towards messenger RNAs. This doesn't have to mean that the RNA is destroyed. It might be inhibited. It might be just localized somewhere else in the cells. But generally, we see in aging, I think you asked, the first question was...
Yeah, so what sort of role does it play in aging?
What we see in aging generally is that microRNAs tend to overall go down with age. What influence exactly this has is not clear, although we did show some studies where microRNAs were shown to prolong or shorten lifespan in C. elegans. But overall, this is quite a good potential. Basically, microRNAs seem to work quite well as biomarkers, for example, and it's already been shown in multiple diseases, such as frontotemporal dementia, and also, I think, some cases of cancer where it functions quite a good biomarker, because microRNAs are generally present at high levels also in the blood, so they're basically quite easily accessible. The other thing to say is, there are also the other things that we discussed, for example, RNA splicing, which also could be a potential target. Maybe there you might be aware that last year in 2019, the antisense oligonucleotides that interfere with the splicing process can be used for specific patients, such as for Duchenne muscular dystrophy, and actually interfere with the spliceosome and allow people with really bad mutations that interfere with the production of this protein to generate the fully functional protein despite that mutation and function normally. These are all potential therapies. Obviously, the antisense oligonucleotides have to be targeted towards single genes. Ideally, we would develop therapies that are focused more generally, and there is where microRNAs do have an advantage, because microRNAs generally have up to hundreds of targets. So if you make a good mix of a single or just a few microRNAs that overall tend to improve your health span, then you might have a quite good turnout. And actually, microRNAs are in clinical trials for heart failure, wound healing, again, some types of cancer. So let's see what comes out of this.
Yeah, I completely agree. We spent some time in class learning about the antisense oligonucleotides in their treatment of muscular dystrophy, and it's a really fascinating application of the technology. So I implore everyone who's listening to go and check that out. That's really interesting stuff. So what do you believe is the next step in investigating the impact of the transcriptome and RNA on aging? And maybe another way to ask this is, if you could design and carry out an experiment right now in this area, what might it look like?
I think with the transcriptome, it's very complicated. There are many things that are dysregulated, and we see that with the proteome and with also the chromatin. There are many aspects. But obviously, I think the most interesting question is all of this really detrimental? There are going to be some causal effects that are going to damage, potential damaging effects, but others might be protective and actually responses to those damaging effects, such as what I described is the downregulation of the ribosome, the protein synthesis machinery. This is a hallmark that actually might be a response and might be actually useful. So obviously we wouldn't want to get rid of some of these factors. So for me, really the question is the hierarchy. Although I did mention that aging might be a collection of agings, as described the CEO of Age Curve, but they might interact, and there is a certain hierarchy. And so, for example, if you get your telomeres shortening, you might initiate senescence, which itself is another hallmark of aging. And too many senescent cells will interfere with cell-to-cell signaling between cells. So that's then going to cause another hallmark of aging. So this kind of progression, I think, is going to be quite common, and it's going to be also present in the transcriptome. Looking at RNA-based regulation, I realized that a lot of RNA, the things we see in RNA might be coming, for example, from the epigenome. So basically what I'd like to do is to look at multiple things in single cells, both the protein, both the chromatin, and also the RNA, and then study tissues through multiple time points and see which changes arise first and how they trigger others. And is there some sequence of events that we can interfere with?
So what I understand what you're searching for is, I guess, what every aging researcher is searching for. It's that root cause of aging. How far up in the hierarchy can you go where you get to a point where you can say, okay, that's a root cause of aging? But I guess that's almost the holy grail at the moment. So my last and final question is understanding how aging affects RNA and transcription may help us design better biomarkers of aging and possibly have clinical relevant endpoints for diseases of aging. Is that a correct assessment? And if so, do you envisage progress towards this objective over the next decade?
Yeah, absolutely. I agree. RNA will be key. And obviously, we already have a good biomarker, which is, for example, the epigenetic aging clock. But to understand more about aging, we would need to sharpen this clock, and potentially by including also the transcriptomic clock and new subtypes, such as the circular RNAs, which can be found in extracellular fluids, such as the blood, could improve its accuracy. So microRNA biomarkers definitely would allow us to that. And also potentially we could link different biomarkers to different hallmarks of aging. So it might be that different people will age differently, having more of an effect on one side, or the more effect on the mitochondria, whereas other people might suffer more from telomere attrition and so on. But personally, regarding RNA, I am really intrigued more and more how much will epigenetics have a role. And during studying for this review, I realized how many links there are. And for example, the elongation rate itself, how fast a polymerase reads the DNA to make RNA can affect basically the capping of the RNA. It's a chemical process, or its splicing, or also which polyadenylation site is used, which final site is selected. And obviously, this rate of elongation can be very much affected by the chromatin, the way a cell is folded. And more this seems to be coming up. For example, in David Sinclair, quite famous ice mice that have numbers of DNA mutations, but they claim, they've assured that these DNA mutations don't essentially cause some damage and are repaired. There is a massive change in RNA splicing due to these epigenomic alternations. And more RNA splicing is coming up as this process. I'll be quite interested what happens if we reprogram cells, if we express Yamanaka factors, bring these damaged organisms or cells back to their youthful state. Is that splicing also repaired? And is it just dependent epigenetic reprogramming? So obviously there'll be a lot of things involved, but surely there'll be some more low-hanging fruits, some of the bigger drivers that we should focus more of our research and the initial therapies.
Amazing. Thank you so much again for joining us. It's been absolutely excellent to hear all your answers.
Thank you so much.
If you enjoyed that, please go and check out our website, www.gowinglife.com, where you can find out loads of articles about all of the news in the longevity world. So again, thank you for listening and goodbye.